I would like to introduce to you Dr. Michael Aschner. Okay. Dr. Aschner is a professor of pediatrics, pharmacology, and senior investigator at the Kennedy Center of Research on Human Development. Dr. Aschner's research interest is on the interaction between genetic and the environment in triggering disease, both during CNS development and senescence. He has been a member or on a board of many organizations and review committees with numerous peer-reviewed publications. Please give a warm Academy welcome to Dr. Aschner.
Good morning, everyone. Before I start, I have to read a disclaimer. So, I do not have any financial interest of the products in my talk or with any company that's offering grant money for this continuing dental and medical education program. I want to take this opportunity and thank especially Rich Fisher, who's in the back, for inviting me again. I was here, I believe it was last year actually, last time that I was here. Okay, so my talk will be about the role of mitochondria in mercury toxicity, and I'll talk a little bit about actually different mercury compounds, but I'll try to focus mostly on mercury vapor and make reference to it because I know it's the most interesting to you. I had an opportunity a few years ago to be in Minamata, which is very famous because of exposure to the methylmercury, another species of mercury, and it's actually eye-opening when you go there and you see a lot of the patients that are still around from the poisoning in the 1960s. There's a museum there, which is here, and there's all kinds of exhibits in the museums and all kinds of pictures. Some of you might be very familiar with. There's also a lot of interesting anecdotal stories that you can learn there. One that was fascinating to me was that the fishermen actually used to moor their boats where the effluent from the factory was flowing that had the methylmercury, because they realized that when the boats were mooring or docking downstream the effluent, they wouldn't have to clean the bottom of the boats because the mercury was so toxic that none of the algae or any of other vegetation would actually stick to the boats. So they just always put it in the same place, which was really interesting. Okay, let's see if I can move this. Okay, now there are a couple movies here. I don't know if everybody wants to watch them. There's some animals here and some humans, but I think it does emphasize how toxic mercury can be. And this is actually a movie that's shown in Minamata, and this is associated with mercury. I think most of you are probably familiar with the story. I can't get it started. Okay, I may have to skip it. But actually the first thing that the people noticed in Minamata was not that people were sick, but they noticed that a lot of the animals were sick. And this first movie actually shows the cats in the Minamata area, which were swaying. They lost their sensory input. They had a lot of motor dysfunction. And in the movie you can actually see a cat on the table. It has absolutely no perception of space. It basically just falls off the table because it basically doesn't know where it is. This is another movie, and I don't know if this one will show. I don't think it will. Okay, this is about another epidemic. I may not have loaded the movies, although it looks like they're here. It doesn't matter. People will probably be happier not to see it after breakfast anyway. But this one shows patients that have been exposed to methylmercury in Iraq, where there was another poisoning. A grain was shipped from Mexico to Iraq. The methylmercury was used as a fungicide, and when the grain actually arrived in Iraq, and this is in the 1970s, it was too late for the planting season. So rather than waiting for 6 or 9 months, what the farmers decided to do, or the people, they decided to use the grain and bake their traditional bread, the pita bread that you're familiar with. And people consumed the methylmercury in bread, and there was a huge epidemic. About 5,000 people actually died from methylmercury poisoning. So I'm going to skip this one because you have the learning objectives. I'll talk intermittently about all different species of mercury, and I'll focus my stuff on mitochondria and some genetic interaction between mercury and neurodegeneration, which is the area that my lab is working in. So just as a refresher, Mercury has 3 different phases. The first one, as everybody here is familiar with, is the mercury from amalgam. There are a couple other issues that are very important. The first one is thimerosal. Thimerosal was used for many, many years in the United States. It's actually still used in some things. It's an organic form of mercury, unlike the mercury vapor. Thimerosal is basically 50% ethyl mercury and 50% other things. And it's used as a preservative mostly in vaccines. Unfortunately, we still use it because it's much, much cheaper to ship the vaccines to foreign countries in big vials rather than have a single vial for every child. And the reason that the ethyl mercury is used basically because it's a wonderful bactericide. It kills bacteria. So the pharmaceutical companies like to have a big vial with a lot of ethyl mercury in there to ship it to third world countries where the doctors can then pull the vaccines and immunize many, many children rather than sending every child his or her own vial. So there's a lot of concern about ethyl mercury, about thimerosal. It was actually removed from vaccines in the United States. I don't know if you know. It's been revisited many, many times, but there are some nasal sprays, for example, that still have thimerosal because it's a very cheap and very effective bactericide. And the last one is methylmercury, which I'll talk a little bit when I talk about our own research. The main concern about methylmercury is obviously from natural exposures associated with fish consumption. And there are many, many populations around the globe that still consume most of their proteins is derived basically from fish, so there are many populations that consume large amounts of methylmercury, which is basically just driven by their diet. This is the mercury cycle. We obviously did not invent mercury. Mercury is in the soil. It's a very abundant metal. We are responsible for a lot of the emission of the mercury, mostly in the form of mercury vapor. It's anthropogenic, it's man-made. And what happens with mercury, actually it cycles in the atmosphere and then it precipitates. When it precipitates, the inorganic mercury is methylated by different bacteria because it's an own defense mechanism for this bacteria. By way of methylating it, they are able to excrete it back into the water, and then the methylmercury starts accumulating in fish animals, in fish and other biota in water. And as you move higher and higher in the hierarchy of marine animals, the methylmercury tends to bioconcentrate. So the way that people consume the mercury, which may originate as mercury vapor, is by this methylation process, which then accumulates in the fish, and the fish are then consumed by human populations. Mercury can travel very long distance. In the 1970s and 80s, the mercury in Europe, for example, was traced from coal burning in the Midwest in the United States. And it's trivia again, but the concentration of mercury in the atmosphere in the northern hemisphere is about 3 times higher than in the southern hemisphere. And the reason for that is because the northern hemisphere is just more industrialized. So if you live in the northern hemisphere, as most of us do here, there's more mercury compared to the southern hemisphere. Now, these are some of the clinical and toxicological features of mercury. And again, I don't think I have to dwell about this too much. The mercury vapor is here. This is the variable. I think you have the slides. The inorganic divalent mercury is here, the methyl mercury here, and the ethyl mercury is here. And I think all of you know the system or the different systems that are associated or sensitive to the mercury vapor. It's largely the central nervous system, but it's not just the central nervous system. There are effects on the lung, depending on the dose, the gastrointestinal tract, the kidney, basically every system, the immune system, is affected by the inorganic or the mercury vapor. Okay, now the different mercury compounds actually have very different effects, and it depends very much on their metabolism or on the species of the mercury. So we have three different species we mentioned. The first one is the mercury vapor. The vapor is by definition inhaled. This is the air, so it can get transported across the alveolar membranes within the lung. It gets into the blood, and then in the blood it gets oxidized to Hg++, to the divalent mercury, inorganic mercury. And this happens in the red blood cells. This process is not very rapid, so there's always enough mercury vapor to diffuse across other membranes, and one of these membranes is the membranes of the blood vessels in the brain. So the mercury vapor can readily cross the membranes of the blood-brain barrier, and therefore it can very readily get into the nervous system. So it's affected by the nervous system. And what happens in the nervous system is, again, the mercury vapor gets oxidized to Hg++. Hg++ doesn't have very good means of getting transported from the brain out, so basically it just gets stuck. And that's how you get all the symptoms in the central nervous system. Now, it's actually very interesting, again, a trivial story, but I was in a mercury mine about 20 years ago in what used to be Yugoslavia at the time, and I learned that the mercury miners were actually drinking a lot of alcohol, and I asked why, and they said, well, when we drink alcohol, we feel much better. And the reason for that is actually scientific, because the mercury vapor gets oxidized to Ag++ by an enzyme that's known as catalase. And alcohol is also a substrate for catalase. So when you have alcohol in the blood, what happens is a lot more of the mercury vapor does not get oxidized to the Ag++, and therefore you tend to also exhale much more of the mercury. So in the presence of alcohol, the body burden of the mercury vapor and the mercury in the body tends to be lower. Now this is a different species. This is inorganic mercury. What happens with inorganic mercury is obviously we ingest it. It gets through the portal vein, it gets to the liver, it binds to glutathione, which I'll get back to, which is a very important antioxidant. And then most of the mercury in the inorganic form actually ends up in the kidney. Not until too long ago, inorganic mercury in the form of mercilil, some of you may have heard about it, was used as a diuretic because mercury is a very potent inhibitor of ATPases, so it was blocking the pumps. So sodium was not absorbed in the kidney, more sodium was in the kidney tubule, and therefore less water was retained. So it was a very effective diuretic, and of course it's not being used anymore. The third species is the mercury in the form of methylmercury. And what happens with methylmercury, again, we eat fish, it gets into the intestinal tract, gets into the portal circulation, gets into the blood, the general circulation, and then, as I'll show you in the next couple slides, mercury binds to thiols, to sulfhydryl groups in the blood, and it has a specific transporter, which is the neutral amino acid transporter, and this is important because it's the means of transporting the methylmercury into the brain. So the methylmercury, mercury vapor affect primarily the central nervous system, whereas the inorganic mercury in the form of Hg++ primarily affects the kidney. And it's driven by the chemical properties of the different mercury species. This is just to show the transporter. So this is it. It has a heavy chain, and I'm not going to go into too many details because I don't think it's that important. There's a couple loops here. And this transporter, the large amino acid transporter, transports amino acids such as methionine. And what happens is when methylmercury, for example, binds to methionine, it has a structure which is very similar to the methionine. So if you wish, the methylmercury cysteine conjugate basically has a mimicry in structure of the cysteine, and this is the means by which it's being recognized by this neutral amino acid transporter, and this is the way through which it gains entrance into the central nervous system. Okay, so I'll talk a little bit now about the mitochondria. And again, you have these slides. I'm not going to give you too many details, but I think what's important to remember from this talk is that the mitochondria are critical targets of the different species of mercury. And I'll show you some about ethyl mercury, I'll show you some about methyl mercury. I haven't done any work with mercury vapor, but basically the same things happens with any of these mercury compounds. So this is the inner membrane of the mitochondria. This is the outer membrane of mitochondria. The mitochondria are obviously where all the energetics is being produced for the cell. And there's a high energy electron that's passed along this electron chain. And the energy that's released pumps the hydrogen out of the matrix. It goes into the space that's between the inner and the outer membrane. And then this gradient, which is created by this, drives the hydrogen back through the membrane, through the inner membrane. And here we have a process where ADP is converted to ATP. So the electron transport chain is basically responsible for the production of ATP. This is what happens normally. Now when you have mercury, this is again the mitochondria, the whole process is basically screwed up. First of all, you get much more oxidative stress. You generate oxidative stress, which is basically electrophilic compounds. These are molecules that are within the cells that are very unhappy. They're not electron neutral, so they're always looking for friends. And when they do that, they tend to extract electrons from proteins, from DNA, from a lot of other things. And in the process, and it's like a brush fire. Once it starts, it just doesn't stop. So you generate different kinds of oxidative stress. In this case, we have a hydroxyl radical. This process happens actually all the time. But when you have mercury on board, it happens much faster, and you have a lot more oxidative stress in the form of sulfhydryl radicals. Now, under normal conditions, we have an enzyme that's known as SOD, or superoxide dismutase, which can dismutate the superoxide with hydrogen peroxide to form water, and the cell can eliminate those free radicals. But when you have mercury on board, this does not happen because a lot of the mercury binds directly to glutathione. Mercury loves glutathione, it loves sulfhydryl groups. So, and the glutathione is the most important antioxidant that we have. So when the mercury binds to the glutathione, there's less glutathione that's available. You get more oxidative stress. You get more iron in the cell. Iron can cause by itself a lot of oxidative stress and cause the production of hydroxyl radical. And as I said, it's like a brush fire, it doesn't stop. Mercury also inhibits a couple of the enzymes that are important in the recycling of glutathione, in the formation of glutathione. One of them is the glutathione peroxidase, and one of them is glutathione reductase. So you can basically see that when you have the mercury on board, when it affects the mitochondrion, the end result is that you're left with less of the antioxidant systems, less of a defense system with which the cell can protect itself from the mercury, and you also generate a lot of toxic species in the form of superoxide and hydroxyl radicals. Now this can be measured. There are different means to do that. And the next few slides just show changes in mitochondrial membrane potential and reactive oxygen species generation. Y-axis is basically the amount. The x-axis is different concentration of mercury. In this case, it's ethyl mercury. But you can see that with the ethyl mercury, you get increased production of reactive oxygen species. This is concentration dependent. Over here we have a time, going from 0 to 60 minutes. You see the same thing, increased reactive oxygen species production, and you're losing the membrane potential in the mitochondria. And that's important because when you lose membrane potential in the mitochondria is not able to generate enough energy to move ions across the different membranes. So when you're dissipating the membrane potential, eventually the cell is just going to collapse. You can also look at it visually. Again, this is the ethyl mercury. I'm not going to say very much about it. The red is what we call a mitotracker, which is a label for the mitochondria. The green is the reactive oxygen species. And this is the combination of the two. If you take these two and put them together, these are the control cells. These are cells that have been treated with ethyl mercury. And you can see that the merging of the color is much stronger when you have the ethyl mercury on board. Again, suggesting that in the presence of ethyl mercury, you're getting much higher rates of oxidative stress. And this is localized to the mitochondria. Carbonyls are another form of measure of oxidative stress. These are carbonyls that are adducts of different proteins. You can look at it also directly, and basically you see the same type of image. And if you look specifically at mitochondrial DNA, again, you could see that in the presence of ethyl mercury, you get much higher production of hydroxyl radicals within the mitochondria, and you get damage to the DNA. So these are the controls. These are the cells that are treated. And when you look at the merging of the different colors, which is indicative of mitochondrial DNA, you can see that it's much stronger in the cells that are treated with a thimerosal compared to the control cells. Again, indicative of mitochondrial DNA damage. So what does it all mean? Or what's the proposed mechanism? So mercury is a lipophilic cation in the case of ethyl mercury. This is the outer membrane, this is the plasma membrane, this is the outside, this is the inside. It will get into the cell because it readily crosses the membrane. The same happens for mercury vapor. Actually, it doesn't need a carrier or anything, it just gets, going the wrong way, it just gets across. It also gets into the mitochondria. This is the outer membrane. This is the inner membrane. Given that there's a very large difference in the mitochondrial membrane potential, so the mercury gets into the mitochondria. As I said before, mercury has very high affinity for thiols, and what it does, it will bind to different thiols on iron clusters, and in the process what it does, it releases the iron. And I've mentioned already, iron is a very potent pro-oxidant through the Haber-Weiss reaction, and then the iron can combine with superoxide and other molecules and form high levels of reactive oxygen species within the cells. And when this happens, obviously the cell will eventually die, because then you're dissipating membrane potential. It's been shown that the mercury can induce the release of cytochrome C from the mitochondria, and it can also induce cell death, what we call apoptosis. So it's a very potent toxin that works through this machinery, basically killing the cells by way of affecting the mitochondria. Okay, now my lab works a lot with worms, and I think you may have seen a couple of these slides last year. We work with worms because they're a wonderful model to look at the interaction between mercury and the genetic of an individual. In this case, we're talking about worms. A lot is known about these animals. They're very small. They're about 1 to 2 millimeters in size. We can freeze them, and we can generate a lot of different mutants. We can knock in and knock out different kinds of genes. So we can ask questions about what's the relationship between mercury exposure and the genetic composition of an animal or of a human. And it's important because even when we're exposed to mercury, each of us is going to react in a different way, and it's driven by our genetic composition. So some of us are more susceptible to it, some of us are less susceptible to it, and this depends on how you handle the mercury in terms of the metabolism, the absorption, what kind of molecules are being affected by the mercury, and basically what kinds of proteins are being expressed, and that's dependent on your genes. And I'll skip some of these. So one of the things that my lab has been interested in is the relationship between the methylmercury and Parkinson's disease. And one day I'd love to look actually at mercury vapor as well and see the relationship with Parkinson's disease. There is some epidemiology and support of a relationship between mercury and Parkinson's disease. So both Parkinson's disease and mercury, this should be an A, I'm sorry, may present with resting tremor and alterations in motor function. As everybody knows, this is true also for mercury vapor. There are a number of studies that have shown that there is an association between fish consumption, which is the methylmercury, and occupational exposure to mercury compounds as well, with an increased prevalence of Parkinson's disease. And it's been also looked at people that have Parkinson's disease, and it's been shown that people that do have Parkinson's disease tend to have higher levels of mercury. So it's not necessarily, it's sort of the chicken and the egg. We don't know that the mercury causes the Parkinson's disease. It's certainly possible that people that have Parkinson's disease for some reason have higher levels of mercury. But I think in general, what we see from these bullets is that there is support for the notion that there is a relationship or association between exposure to different species of mercury and Parkinson's disease. Now, one of the genes that's important in Parkinson's disease, people that have mutations in this gene, which is known as PARK2 or Parkin, is people with this mutation have nearly 50% of familial PD cases are associated with mutations in this gene. The protein product of this gene is known as Parkin or PDR1 in the worms. And I'll tell you about the function of this in just a minute. It's what we call an E3 ubiquitin ligase. The important thing to know, again, that it's a risk factor for Parkinson's disease. So from your perspective, it's obviously important to know if a patient that has a mutation in this gene, and there's about 10, 12 other genes that have been shown to be associated with Parkinson's disease, is this kind of patient going to be more sensitive to the mercury vapor? And the likelihood for this is yes. So what we did in this case is we examined the impact of mercury exposure, and in this case it's methylmercury, on the mutants of this gene, PARK2, Parkin, or as we call it, PDR1. And we basically tested the hypothesis that animals, in this case the worms, that have mutations in this gene are going to be more sensitive to the mercury. So in your case, the question would be again, is a person that has a mutation who doesn't show actually any symptoms, because as you know, the non-familial Parkinson's disease does not develop until about the age of 60, 65. So if you have a patient that's 25 or 30 years of age, they may have a mutation in this gene. They don't have any behavioral or clinical symptoms of Parkinson's disease, but they may be predisposed to develop Parkinson's disease. The question is, are they going to be more sensitive, or by being exposed to mercury from amalgams, you don't use the mercury, but other dentists should consider whether being exposed to mercury, they may hasten the disease process. And as I said, there are probably about 10, 15 other genes that have been associated with Parkinson's disease. So I'm going to go through these slides pretty quickly. The first thing that we did was to look at the number of live worms. It's sort of life dead. I mean, are we killing more worms as we increase the concentration of mercury? And are the mutants, the PDR1 knockouts, the animals that don't express this gene, are they going to be more sensitive to mercury? And the answer is yes. So this is a typical dose response. As you increase the dose, you start with 100% live worms, and as the worms get exposed to higher doses, you can see more of them die. Eventually, if you use enough mercury, they all die. What you see here in the hatched line is that if the animals are knockouts of this gene, they're more sensitive. If you compare this dot here to this dot here, you need less mercury here to cause 50% death. So the LD50, the lethal dose 50 in the PDR knockouts is lower compared to the wild types, compared to the control. So animals that have mutation in this gene, or in this case where the gene has been knocked out, are more sensitive to mercury. Again, the question is, does a patient that have a mutation in PDR1 or Parkin, when they are exposed to mercury, are they going to be more sensitive? It might not be life and death, because this is pretty acute experiment, but over a period of 30 or 40 years, if they're exposed to the mercury, are they going to be more sensitive than an individual that doesn't have this mutation? And the answer is probably yes. We also looked at the survival. This is basically a lifespan. And again, without too many details, you could see that in the N2, which are the control animals, the more mercury you give them, and the more sensitive they become. There's a shift to the left. So lower dose, again, is causing an LD50. But you can see that the animals that are mutants, or the knockouts, are much more sensitive. You can compare this line here to this line here. And you can see that when you give them the mercury, there's actually a greater separation of these lines. So they're much, much more sensitive to the mercury. And it's pretty significant, because an animal that is not exposed to mercury, a worm, lives about 27, 28 days. If you take an animal that's a mutant and you give them the methylmercury at 20 micromolar, which is not a very high concentration, they live only 17 days. So you almost cut about 60%, 80% of their lifespan by exposing them to mercury. And again, this does not happen necessarily in humans, because these are pretty acute experiments, but I think it does show how sensitive these animals are to the methylmercury. We also looked at the production, we looked at the amount of mercury in these worms. And what we found very interesting is the animals that are the knockouts of this gene actually tend to accumulate a lot more mercury at each of the doses. This is 10, and it's significant at 20 micromolar. So the question was why? And as I mentioned before, Parkin is an E3 ligase. The function of Parkin is basically to take different proteins, to ubiquitinate them. They put a ubiquitin group on these proteins, and then when these proteins are being ubiquitinated, it's hard to say, they are degraded in the proteasome. So if you have a mutant E3 ligase, if it doesn't work, this process is obviously not working as it should. So what we decided to do was to see whether the animals that are mutant, who have more mercury in them, also have higher expression of the transporter for mercury. The idea was that the neutral amino acid transporter simply won't be ubiquitinated, and therefore it wouldn't be presented to the proteasome, and therefore the levels of this transporter would be higher, which would translate to having more mercury coming into the worm. And we did that. And there are three different actually isoforms of this large amino acid transporter. It's called AAT1, 2, and 3 in the worm. And you can see that for two of these isoforms, AAT1 and AAT2, when you knock out this gene, the PDR1, the Parkin, they tend to have more transporter. So it fits very well. They're more sensitive, they have more mercury, and the reason that they have more mercury is that they have more transporter in the membrane. So the mercury can come into the cells much more readily. We also looked at oxidative stress, and we do find an effect of mercury, but we were surprised actually to find out that the amount of oxidative stress was not different between the control animals and the mutant ones, the ones where we have knocked out the gene. But we weren't deterred, and the question that we asked was, well, maybe there's the same amount of oxidative stress in these animals, but maybe their ability to respond to the oxidative stress is very different. So we decided to look at the system that's responsible for regulating and handling the excess of the oxidative stress. And this is done by a system that's known as NRF2 or SKN1 in the worms. Basically, what we have in our system is, under normal condition, we have NRF2 in the cytoplasm, and it's bind to another protein which is known as KEAP1, And then when we have oxidative stress, the keep one has some sulfhydryls on it. These sulfhydryls get oxidized by the oxidative stress. The keep one dissociates from the NRF2, and then the NRF2 moves into the nucleus. It binds to an antioxidant response element, and you get upregulation of all kinds of different enzymes that are responsible for reducing, attenuating the oxidative stress. So our question was, is this system actually functional to the same degree in the mutants compared to the controls? So we did that, and as we expected, we get a very robust response in the animals that are controls when we treat them with methylmercury, but in the animals that are mutant, they don't express the PDR1, there's a very blunted response compared to the wild-type animals. So there's more mercury coming in, there's more protein that brings the mercury in, the animals are more sensitive, and the reason for that is that for the same amount of oxidative stress, they are not able to respond at the same level. So the inference from this, obviously, is that there's going to be much more oxidative stress and much more damage, which translates to the reduced lifespan, and the reduced viability, and so forth. And this is here, I'll skip that. The next thing that we decided to do was to look at the relationship between the ability to respond to oxidative stress. Again, we're talking about the same system, NRF2 or SKN, and methylmercury. And the rationale was that animals that do not express NRF2 to the same level as a normal individual would be more susceptible to the mercury, because we know that one of the mechanisms of mercury-induced toxicity is by way of oxidative stress, so you need the NRF2 system, and by production of free radicals. So the question is, if animals are deficient in this system, are they going to be more sensitive, because you're going to get much more oxidative stress, effects on mitochondria, as we discussed before, and all these types of functional things that I've described before. So this is basically the same curve that I've shown you before. This is the number of live worms. This is the concentration of mercury. The blue one is the controls. Again, as you increase the concentration of exposure, more worms die. And you can see when we look at the SKN1 knockouts, the animals that have a deficient NRF2 response, they're much more sensitive to the mercury. There's a shift to the left, which makes perfect sense. Again, if you translate it to a patient, if you look at the NRF2 system in 1,000 people, we are all different. Some of us are what I call good responders to oxidative stress, and some of us are bad responders to oxidative stress, because we express different variants of the NRF2, different alleles of the NRF2. So some of us can tolerate oxidative stress to a much greater extent than other people. And the question becomes then, if somebody has a mutation in the RNF2, are they going to be more sensitive to the mercury? And you can even think about it in other terms. If somebody has a mutated or mutant NRF2, and they also have one of the genes that associated, or a mutation in one of the genes that's associated with Parkinson's disease, the question becomes, do these two systems actually synergize? Is it additive? Is it multiplicative? It could be a tenfold difference in terms of sensitivity to mercury and many other things. So it becomes quite significant. The lifespan, again, is affected in SKN1 knockouts. These are the controls. Interestingly, the ones that are not exposed live about 27 days, but you could see once you, I'm sorry, these are the N2s, these are not the mutant animals. The animals that have a deficiency in SKN1 live shorter, just without exposure to mercury. And then you can see when you expose them to mercury, you get a further shift to the left, and you see the same thing with animals that are exposed to mercury and are wild type, so they don't have the mutation. But you can see that with the mutation, they're much more sensitive to the methylmercury. You see the same thing in terms of reactive oxygen species, a much greater reactive oxygen species production in the animals that are mutants of the SKN1. You see more mercury accumulating in these worms. And what we also do specifically is we look at dopaminergic neurons. I didn't show you any movies of these worms, but they're basically transparent. And what we can do is we can label different neurons with specific fluorescent markers. So we can count how many neurons are in the worm and then before and after exposure to mercury. And what we find is with animals that are mutated, that have a mutation in SKN1, the number of dopaminergic neurons and the dopamine, the content of the dopamine in these animals is significantly reduced to the wild types, and this is shown here. So these are the wild types. These are the different neurons with a red cherry marker. When they're exposed to mercury, the dopamine disappears, which is very, very significant because, as you know, the dopamine is basically the neurotransmitter that's associated, or the loss of dopamine is associated with Parkinson's disease. These are the mutants. They do have dopamine neurons that you'd expect, and you can see that with the mercury exposure, the dopaminergic neurons almost completely disappear. Again, consistent with the fact that these animals are much more susceptible. So I think the idea is, again, to think about the interaction between the genetics and the exposure, because some individuals are definitely much more sensitive to the mercury, given that they may carry mutations in some of the genes that are associated with neuroprotection or with making them more susceptible to different neurological disorders. And I would say it probably translates to any other disease as well, in the kidney, in the gut, immune responses, and so forth. It's not just the nervous system. I'll skip a few of these because I think I'm running out of time. Okay, I want to finish up by talking again about the mitochondria. And I talked about it before. One of the first slides, I showed you the production of ATP. I showed you the hydrogen moving along the respiratory chain and the gradient and how the hydrogen is being pumped out. In the process, the ADP transforms to ATP. What you need for that is what has been termed a universal cofactor, nicotinamide adenine dinucleotide, or NAD. That's why it's abbreviated, so we won't have to say it. As an essential coenzyme, NAD gains 2 electrons and a proton from substrates at multiple the tricyclic acid cycle steps, and it's being reduced to NADH. Mitochondrial NADH is oxidized upon donating its electron to complex 1. The NADH ubiquinone oxidoreductase of the electron transport chain. And then, as I've showed you before, it can move along the different complexes, and it's important in the production of ATP. And this is just shown schematically here. The NAD has different kinds of functions. This is the electron transport chain, and you can see the NADH in the various processes, and the TCA cycle, which is important for the energetics within the mitochondria. Now, these are the dopamine neurons. This is in the worm, and again, I won't give you too many details. But basically, these worms have the machinery that we have in mammalian systems. It's essentially identical. So we can look at these neurons, and it's not going to work, as I've shown you in the previous couple slides, in the different animals. And again, this is cherry red. These are just different types of neurons, which are known as ADE, CEP. These are the axons of these neurons. These are the controls. These are animals that have been exposed to mercury, and you can see that you lose fluorescence, which is indicative of the fact that these neurons are disappearing. These are animals that have been treated with NAD, which is an important cofactor, so we're basically trying to protect the worms by giving them NAD. There's no difference between these neurons and these neurons. And what's pretty amazing is that you can rescue the damage that you see with the mercury when you give the animals NAD. So, and I think there have been some talks actually last year. I remember listening to different talks about rescuing mitochondrial function and how it might be important in rescuing some of the damage that's associated with mercury. But in this case, you can recover the loss of dopaminergic innervation in mercury-treated animals by supplementing them with NAD. This is NAD in nanograms per milligram protein, and you can see that when you treat with mercury, the amount of NAD goes down with 10 and 20 micromolar of mercury, goes significantly down, and when you supplement with NAD, actually the levels go up, and it's consistent with what I've shown you in the previous slide. When you have enough NAD, you can rescue this dopaminergic neurodegeneration. In this case, NAD prevented the mercury-induced changes in a basal slowing response. Basic slowing response is basically just a behavior in the animals. When the animals are exposed to food, they tend to slow down, which makes sense. You know, we may run to McDonald's, but once we get to McDonald's, we sit down and we eat. So the worms do basically the same. They look for the food. When you give them the food, the bacteria, they slow down. If you give them mercury, there's an effect. You can see there's a change in body bends. They move much slower. So the mercury affects their motor function. If you give them NAD, again, you can recover their motor function. This is a label for the mitochondria. So this looks at membrane potential of mitochondria in the whole worm. And when you lose membrane potential, these animals become fluorescent, much more fluorescent. So this is without mercury. This is with 10 and 20 micromolar mercury. You could see these animals are very stressed compared to their controls. And if you give them NAD without mercury, they look the same. And if you supplement the animals with NAD, you can rescue them again from the effect of the mercury on the mitochondria. And this is quantified here. You see basically the same thing. We also looked at the ability of NAD to protect against glutathione depletion. As I mentioned before, glutathione is the most important antioxidant. When you treat animals with mercury, you lose glutathione significantly, both with 10 and 20 micromolar. And when you give them NAD, you can recover the glutathione content. And this is again because NAD is important in the mitochondrial function and the recycling of glutathione through glutathione peroxide and glutathione reductase, as I've shown you in one of the earlier studies. So, in summary, I hope that I established to the best of my ability the centrality of aberrant mitochondrial function in mercury-induced effects, and it's not just unique to dopaminergic neurodegeneration. I've also shown you effects on viability and other stress functions within these animals. Once we lose NAD, we see altered mitochondrial function, we see loss of membrane potential, loss of glutathione, loss of motor function, a lot of other deficits in these animals. And we also see, importantly, because from my perspective at least, the interest was in dopaminergic degeneration, Parkinson's disease, we see that these animals have reduced content of dopamine within their neurons. And this can all be rescued by giving them exogenous NAD. So these mercury-induced effects are attenuated by NAD supplementation, and basically, hopefully what I convinced you is that the mitochondria are specifically and exquisitely sensitive to mercury. I talk mostly about methylmercury, but the same thing I'm sure happens with inorganic mercury and with mercury vapor, and these have yet to be tested. But since they are going to get into the mitochondria, I don't have any doubts that you'll see very similar effects. And then we also have to think about how all these things interact with the genetics of an individual. And hopefully I convinced you too that individuals that have mutations in certain genes might be much more susceptible to the effects of mercury. So thank you. And one disclaimer, this was funded by, I don't have any interest in any products, but this was funded by the NIH. And obviously I'm very thankful for their support. Thank you for your attention.
Thank you, Dr. Aschner.
Do I take questions now or 5 o'clock? Okay, thank you.